The topic selected for today is eukaryotic cell. This is part 2 of cell, the unit of life. Eukaryotic cells are present in the kingdoms Protista, Plantae, Animalia and fungi. In the previous class I have told you the difference between prokaryotic and eukaryotic cells. Prokaryotic cells have no nuclear membrane around their chromosome, and the prokaryotic cells are present in Munira. Whereas in the case of eukaryotic cells, the chromosomes are surrounded by a nuclear membrane. This is the main difference between prokaryotes and eukaryotes. Today, let us describe the eukaryotic cells. The eukaryotic cells present in plants and animals have some differences. For much of their parts, they are similar, but in some respects, they differ from each other. In the case of plant cells, we come across a cell wall, a cellulose cell wall, which is absent in animal cells. Then, in the case of plant cells, there are chloroplasts, which are absent in animal cells. Then, in the case of plant cells, there is a large central vacuole. The vacuole occupies much of the cell in the case of plants. In the case of animals, such a large central vacuole is absent, but some small vacuoles like food vacuole, contractile vacuole, etc. are present. But a large vacuole is absent in animal cells. Then, in the cell wall of plants, there are channels known as plasmodesmata. The plasmodesmata connect the cytoplasm of neighboring cells. They are also present in some of the animal cells. Then, in the case of animal cells, there is one cell organelle known as centriole, this one. The centriole is located near the nucleus and is present only in animal cells. Except for very few, plant cells do not have any centriole. The centriole participates in cell division. Then, most of the parts are the same in both plants and animal cells. See, this is the nucleus. Here is also nucleus. The nucleus is surrounded by a double-layered membrane known as nucleolemma. It has a number of openings known as nucleopods. The endoplasmic reticulum is attached to the outer membrane of nuclear membrane. Inside the nucleus there is chromatin, nucleolus. Here also we find chromatin and nucleolus inside the nucleus. The nuclear membrane has pores. Surrounding the nucleus there are ribosomes in both the cases. In both the cases there are mitochondria. In both the cases there is rough endoplasmic reticulum, smooth endoplasmic reticulum, cytoplasm, 80S ribosomes. Then there are Golgi apparatus in both plants and animals. So in this way, the plant and animal cells differ only in some details. Except for this, they are identical and they perform similar functions. Then on the animal cell, we also come across microvilli in some cases. In the case of small intestine, and also in the case of proximal convoluted tubule of nephron, we come across microvilli. Microvilli increase the surface area of absorption. Now, let us take up various organelles present in the eukaryotic cell. In the previous class, I have told you the cell membrane is almost identical in both prokaryotes and eukaryotes. The cell membrane is also known as the plasma membrane or plasma lemma. It has a number of names. The plasma membrane is made of a lipid bilayer. It is made of two layers of lipid molecules. The structure of the plasma membrane was explained by Singer and Nicholson in the year 1972. So with the help of an electron microscope, Singer and Nicholson studied the plasma membrane of RBC. Empty RBC cells are known as ghost cells because RBC have no cell organelles except for hemoglobin. So when we remove the hemoglobin from the RBC, they are called ghost cells and they are very easy to use for studies. So in the ghost cells of RBC, these two scientists have found that the cell membrane is made of a lipid bilayer, and the model proposed by Singer and Nicholson is known as fluid mosaic model. So I have drawn one lipid molecule here. This is another lipid molecule. A lipid molecule has a polar head, then two tails made of fatty acids. The polar head is hydrophilic, that means it is water loving, so it remains outside, towards outside. Whereas the fatty tails are hydrophobic, they hate water, that's why they face inwards. So in the lipid bilayer, the polar heads of the lipid molecules face outside, whereas the fatty acid tails face inside. So this is the arrangement. This is one layer of fatty lipid layer. This is another lipid layer. Then embedded in the lipid layer, there are protein molecules. These are known as integral proteins or embedded proteins. Proteins are also present outside and inside the plasma membrane, and these are known as peripheral proteins. So plasma membrane is mainly made of a lipid bilayer, and it is also associated with integral and external protein molecules. Attached to the protein molecules, there are carbohydrates. Then the lipid bilayer is in a semi-fluid state and exhibits lateral movements. So the protein molecules can also move along with the lipid layer. Then the plasma membrane is semi-permeable and also selectively permeable. It allows only some molecules to enter. Then the plasma membrane exhibits three types of transport. Passive transport, it occurs through osmosis. Water moves from higher potential to lower potential. Then active transport, it involves expenditure of energy. Sodium potassium pump is a good example for participating in active transport. The third one is facilitated diffusion. This is done by a carrier protein present in the lipid bilayer. And facilitated diffusion doesn't involve any expenditure of energy. In this way, the plasma membrane helps in active transport, passive transport, and facilitated diffusion. It forms the outer boundary of the cell. Then, cell wall. Coming to the plants, there is a cell wall surrounding the plasma membrane. The cell wall is non-living. In fungi, it is made of cellulose, galactose, mannans, and salts like calcium carbonate. In plants, the cell wall is made of cellulose, hemicellulose, pectines, and proteinases. Then, when a plant cell is growing, it is first surrounded by a primary cell wall. The primary cell wall allows growth of the cell. Once the growth of the cell is completed, the primary cell wall is replaced by a secondary cell wall, which forms inside the primary cell wall. Then, see here, this is the cell wall of this cell, and this is the cell wall of another cell, neighboring cell. The two cell walls are separated by a middle layer. This is known as this one, this middle layer, and middle layer is made of calcium pectate. It is made of substance known as calcium pectate. Then traversing or passing through the cell wall and middle layer, there are channels, these channels. The channels are known as plasmodesmata. The plasmodesmata allow movement of substances between the neighboring cells. So that is about cell wall. Then coming to the endoplasmic reticulum. Endoplasmic reticulum is a network of tubules or channels, vesicles, and it divides the cytoplasm into luminal and extraluminal space. Luminal means the space enclosed by the endoplasmic reticulum. The space outside the endoplasmic reticulum is known as extraluminal space. The endoplasmic reticulum is actually two types: rough endoplasmic reticulum and smooth endoplasmic reticulum. In the case of rough endoplasmic reticulum, it appears rough in appearance because it is covered by 80S ribosomes. In the case of smooth endoplasmic reticulum, the ribosomes are not attached to its outer surface, so it appears smooth. Then, the endoplasmic reticulum connects the nuclear membrane with the plasma membrane, like this. Then, inside we find a network of rough endoplasmic reticulum. The ribosomes attached to the rough endoplasmic reticulum participate in the synthesis of proteins, enzymes, and hormones, and they are released into the lumen of the rough endoplasmic reticulum. From this lumen, they are released into the cytoplasm in the form of secretory vesicles. These vesicles take the substances into the Golgi apparatus. So in this way, the substances produced are produced by the ribosomes present on the rough endoplasmic reticulum are transported to the Golgi apparatus for processing and repackaging. I will explain that later on. So this is the smooth endoplasmic reticulum. The function is also somewhat different. Rough endoplasmic reticulum participates in protein synthesis mainly. Smooth endoplasmic reticulum participates in the synthesis of steroid hormones and the lipids. So that is the main difference between RER and SER. Then the Golgi apparatus. This is the Golgi apparatus. Golgi apparatus was discovered by Camillo Golgi in the year 1898. It is made of a number of stacked cisternae or sacks like this. They are placed one out like this. So, the Golgi apparatus is made a number of stacked cisternae. I have drawn 3 cisternae here. See, this side, the Golgi apparatus has a convex face. This side it has a concave face. The convex face is known as cis face or forming face. The concave face is known as trans phase or maturing phase. Just as I have told you, the rough endoplasmic reticulum releases substances produced in it through vesicles. These vesicles travel and fuse with the cis face of the Golgi apparatus. They fuse here with the surface. Then the Golgi apparatus is a network of, it produces a number of tubules, and these tubules are interlinked. It forms a maze-like structure. So inside the Golgi apparatus, the substances synthesized in the rough endoplasmic reticulum are modified according to the need of the organism, and they are repackaged again and released into the cytoplasm from the trans phase, like this. And these vesicles are known as secretory vesicles. These secretory vesicles may be released to the outside or they may be used up inside the cell. The secretory enzymes, the secretory vesicles, they contain a variety of enzymes. participating in different activities of the cell. That is about Golgi apparatus. Here I have drawn. Some vesicles travel, fuse with the plasma membrane and release their secretions to the outside. This process is known as exocytosis. Plasma membrane also helps in taking fluids into the cell. This is called drinking. So, if the plasma membrane takes the fluids into the cell, the process is known as endocytosis. And the vesicles formed through endocytosis are known as pinocytic vesicles. Then, coming to the lysosomes. Some of the secretory vesicles form lysosomes. Lysosomes are known as suicidal bags of the cell. That means, if there is a need, they digest the cell itself. The lysosomes are also membrane-bound vesicles, and they carry enzymes known as hydrolases. These hydrolases are acidic in nature, and they participate in the digestion of lipids, proteins, and also carbohydrates. They also participate in autophagy and autolysis. Autophagy means if any of the cell organelles are useless or functionless, they are digested by the lysosomes. When the cell is injured or harmed, or during senescence or old age, the lysosomes dissolve the cell. This is known as autolysis. Autolysis means self-dissolution or self-destruction. In this way, lysosomes participate in various functions, but they are produced by the Golgi apparatus. Then, vacuoles. As I have told you, in plant cells there is a large vacuole which is absent in animal cells. This large vacuole is surrounded by a membrane known as tonoplast. Tonoplast is also selectively permeable. It concentrates unwanted substances into the vacuole through active transport. So the concentration of some of the substances are more inside the vacuole than outside the vacuole. Then, in the animal cells, we come across mainly two types of vacuoles, which are very small in size. They are called contractile vacuoles and food vacuoles. The contractile vacuoles are present in freshwater organisms. They expel the water entering the body through endosmosis. Otherwise, the animal may absorb more and more water and ultimately it bursts and dies. So, to prevent the danger of death, the contractile vacuoles will be expelling all the fresh water that enters their body continuously. Then the food vacuoles. The food vacuoles participate in digestion of the food. Then, mitochondria. Mitochondria are self-replicating cell organelles. The mitochondria are present in cytoplasm. They are surrounded by two membranes. This is the outer membrane, this is the inner membrane. Between the outer membrane and inner membrane, there is a space, narrow space known as intermembranal space. Then, the inner membrane produces inward growths known as the crista. This one, these are all crista or cristae. The cristae increase the inner membrane area by many times. Both the outer membrane and inner membrane have different types of enzymes that participate in Krebs cycle and also in electron transport. Mitochondria are known as power plants or powerhouse of the cell. All the ATP we require is mainly synthesized in the mitochondria. Mitochondria has all the machinery for the synthesis of ATP. ATP is the only source of energy available to all the organisms. Then, the inner membrane includes a space known as matrix. Inside the matrix, we come across a circular DNA molecule or a circular chromosome. Then, inside the matrix there are also phosphate granules, these things, and also 70S ribosomes. So mitochondria has 70S ribosomes. The 70S ribosomes are made of two subparticles. They are 50S and 30S. Then, the shape of the mitochondria is cylindrical or sausage shape. They multiply on their own, and they are the sites of aerobic respiration. Whatever oxygen we consume is utilized by the mitochondria in aerobic respiration or in the Krebs cycle. Then, plastids. Plastids are present only in plants. They are completely absent in animals. The plastids are three types again: chloroplasts, chromatoplasts, and leucoplasts. Chloroplasts contain chlorophyll and they appear greenish and they mainly participate in photosynthesis. Chromatoplasts, they have various pigments, so they appear yellow, yellowish, orange, or reddish in color. Leukocytes is colorless. These are colorless plastids. The leucoplasts are again three types. Amyloplasts, they store starch. Eleoplasts, they store fats and oils. Eleuroplasts, they store proteins. In this way, plastids participate in different functions. Among the plastids, chloroplasts are very important. Chloroplasts and mitochondria were once free-living bacteria, and they ended up as cell organelles in eukaryotes. So, like mitochondria, it is also surrounded by two membranes, outer membrane and inner membrane. Here also there is a circular chromosome, just like mitochondria. The inner membrane encloses a space known as stroma, or a fluid known as stroma. Inside the stroma, we find 70S ribosomes, just like mitochondria. Then, inside the stroma, we come across lipid droplets and also stromal lamellae. We come across thylakoid membranes. These are thylakoid membranes. The thylakoid membranes contain chlorophyll and they are bag-like structures or sac-like structures and arranged one above the other like the piles of coins. These stacks of thylakoid membranes are known as grana, singular granum. This is one granum, this is one granum, this is one granum, like that. Different grana are connected by lamellae known as stromal lamellae. This is one granum, this is another granum. These two are connected by stromal lamellae. So in this way, different grana are interconnected by stromal lamellae. Inside the chloroplast, photosynthesis occurs. Already I have explained the topic. Then, next, ribosomes. In eukaryotes, both 70S and 80S ribosomes are present. Already I have told you, 70S ribosomes are confined to mitochondria and chloroplasts, whereas the 80S ribosomes are present in the cytoplasm. The ribosomes are either free in the cytoplasm or attached to the endoplasmic reticulum, nuclear membrane, etc. The 80S ribosomes are made of two subparticles of 60S and 40S. The ribosomes are factories where proteins are synthesized. Then, cytoskeleton. Inside the cytoplasm, there is a skeleton made of fine tubules. The cytoskeleton not only gives a shape to the cell, it also helps in cell division and in many other activities. It provides mechanical support. It helps in internal transport. It helps in mobility. It provides a definite shape. Like that, cytoskeleton helps the cell in many ways. Then, cilia and flagella. Most of the animal cells have cilia and flagella. Cilia are shorter in length, whereas flagella are longer in length. Cilia are numerous in number, whereas flagella are few in number. But their basic structure is the same. Again, remember, the flagella present in prokaryotes is different from that of the eukaryotes. In eukaryotes, both cilia and flagella have the same structure, and they arise from a basal granule. A basal granule is present just below the plasma membrane. If you take a cross section of the flagellum or cilium, it reveals 9+2 arrangement of microtubules. So there are 9 peripheral tubules. These are peripheral tubules. These are 9 in number. 1, 2, 3, 4, 5, 6, 7, 8, 9. Not only they are peripheral, but they are doublets. Each peripheral tubule is made of 2 subunits. So they are called doublets. In the middle of the flagellum or cilium, you find 2 microtubules. These are known as central tubules. The central tubules are connected by bridges, cross bridges. And they are also surrounded by a membrane known as central sheath. So inner tubules are 2 in number, connected by cross bridges and surrounded by a central sheath. The outer sheath is nothing but plasma membrane. The peripheral fibrils are connected with the central sheath by radial spokes. These are the radial spokes. Then, the peripheral doublets are also joined by linkers. So the peripheral tubules are interlinked by linkers. So this is about the electron microscopic structure of a flagellum or cilium. The flagella and cilia help in locomotion or movement, and in the lower animals they also help in movement from place to place. Then, nucleus. This is the nucleus. Nucleus is well defined in all the eukaryotes. The nucleus is surrounded by a nuclear membrane, which is a double layered structure. This is the inner layer, inner membrane, this is the outer membrane. The outer membrane is connected to the endoplasmic reticulum. Then, the nuclear membrane has a number of small pores through which RNA and proteins pass in and out. The nucleus encloses a fluid known as nucleoplasm. Inside there is a nucleoplasm. And in the nucleoplasm there is a reticulum or chromatin reticulum. Chromatin reticulum is nothing but DNA associated with RNA and also with proteins, histone and non-histone proteins. Then, inside the nucleus, we also come across one nucleolus. Nucleolus is not surrounded by any membrane, and it is the site for synthesis of ribosomal RNA. So it is very important. Nucleolus is not membrane bound and it is involved in rRNA synthesis. Then, the chromatin occurs in 2 forms, euchromatin and heterochromatin. Heterochromatin appears dark in color. Heterochromatin means condensed chromatin. It is condensed and it is not active. It means it is not participating in any transcription process. Euchromatin means dispersed chromatin. Chromatin is dispersed and it is actively participating in transcription. So heterochromatin means condensed chromatin, euchromatin means dispersed chromatin. That is about the structure of the nucleus. Then, inside the nucleus, we come across chromosomes. Chromosomes were discovered by Robert Brown in the year 1891. The number of chromosomes varies from organism to organism. In human beings, there are 46 chromosomes or 23 pairs of chromosomes. Each set of chromosomes is known as a genome. So 23 chromosomes form two genomes in our cells. One genome comes from mother and other genome comes from father. Then the chromosomes have a constriction known as the primary constriction. This one here. Constriction means narrow down. So all the chromosomes have a primary constriction. The primary constriction divides chromosomes into two halves. This is one half, this is another half. Based upon the location of the primary constriction, chromosomes are divided into four types. The first type, metacentric type. The primary constriction is also known as centromere. Don't confuse centromere with centriole. Centrioles are different from centromeres. So the primary constriction is also known as centromere. I have drawn here. This is the primary constriction or centromere. On either side of the centromere, there are kinetochores, this one, the black ones. These are kinetochores. During cell division, the spindle fibers are attached to the kinetochores. So the kinetochores provide surface of attachment to spindle fibers during cell division. If the primary constriction is exactly in the middle of the chromosome, we call that chromosome as a metacentric chromosome. Both arms are equal. This is one arm, this is another arm. Submetacentric. If the centromere is slightly away from the middle of the chromosome, it is known as submetacentric. So here, the centromere is slightly away from the middle part of the chromosome. If the centromere is almost at one end of the chromosome, it is known as axocentric. If the centromere is at the end of the chromosome, if it is at the end of the chromosome, it is known as telocentric. So in this way, based upon the position of the centromere, chromosomes are classified into metacentric, submetacentric, acrocentric and telocentric. Sometimes there will be a secondary constriction. See here, this is secondary constriction. When there is a secondary constriction, satellites are produced. This is satellite. In this way, there are chromosomes situated inside the cell, and chromosomes appear only during cell division. During interphase, we can't see any chromosomes because the chromatin is dispersed. So the chromosomes take their shape only during cell division. Then, in human cells, all cells have all 46 chromosomes. If you measure the length of DNA present in a single cell, it is around 2 meters in length. But it is coiled many times. That's why it can be accommodated within the small space of the nucleus. So that is about the structure of eukaryotic cell. Thank you.